Hello, everyone. Thank you for joining today's seminar. Our first seminar will be given by Guido Barzaghi, who is a PhD student in EMBL and the University of Heidelberg in computational biology. So Guido performed his bachelor degree in theoretical in application of third-generation sequencing method to methylome analysis, where he focused on that for his thesis dissertation. He then obtained a bioinformatics master degree from University of Copenhagen for 2 years, where he studying the on a velocity of single cells in human neurodevelopmental dynamics. Since 2019, he joined EMBL for his PhD degree to focus on how transcription factors kind of establish the chromatin accessibility with the live cell imaging. So he has performed multiple researches, mainly in computational genomics, but he has also participated in many conferences as well as teaching and outreach events, such as teaching in practical for pre-doc course, as well as for some of the summer schools of the single cell, and organize the multiple laboratory treat events, etc. So with that, I would like to hand the floor to Guido to tell us more about his thesis work, where he focused on transcription factors and the chromatin accessibility.
Yeah, thank you very much.
Yeah, I think you.
Yeah. Okay. Well, good afternoon, everyone. Good morning to the ones in the US. Yeah, I would like to first thank the organizers for the chance to talk on this platform. I actually really enjoyed the seminar series throughout my PhD. Okay, so let's get into it. So transcription factors regulate gene expression at cis regulatory elements. And one of the most prominent acts that TFs need to achieve to exert the regulatory function is to actually bind, access and bind their recognition motifs within cis regulatory elements. And for that, chromatin itself is imposing as a physical barrier, creating TF nucleosome competition as a very interesting mode of regulatory activity. So this is perhaps most intuitive when thinking of transcription factor pioneering, where transcription factors are able to access motifs within closed chromatin, allowing for the initiation of a transcriptional program that then leads to cell differentiation. However, something that is perhaps not as frequently thought about is the fact that TF nucleosome competition also exists at the steady state, where transcription factors continuously need to outcompete nucleosomes in order to maintain chromatin accessibility in a homeostatic level, in a physiological level, and then to maintain indeed the levels of transcription that are needed for the cell. So a pretty big reason why understanding the individual contribution of TFs to chromatin accessibility is complicated is because mammalian crees are intrinsically complex elements. They are on average harboring 5 to 6 TF DNA binding events, and this involves transcription factors also combine their actions in ways that can be nonlinear to amount to the final chromatin accessibility of the region. So there can be definitely additive as well as cooperative effects, and there is a pretty widespread degree of redundancy, as we will see in a minute in the system. So there is quite a lot of unknowns of how indeed transcription factors do maintain chromatin accessibility crease. And I would say one of the most interesting questions that we can ask is, what is this quantitative contribution? How much does an individual instance of a TF contribute to chromatin accessibility? And also, how much do they assemble, and how much do does the assembly of TFs contribute to the same? Also, it's quite interesting to ask, in my opinion, how does dendin translates into transcriptional activity? Namely, is like transcription affected by fluctuations in the competition between transcription factors and nucleosomes? So to a complex question comes the rest to a complex technology, which is single molecule footprinting, which is a single molecule genomics assay. And to explain what it is and why we use it, I will go through an example. So say we want to dissect an inactive locus which harbors an OCT4 and SOX2 binding sites, which I am indicating here with this gray dashed line. So if I was to approach this with traditional bulk assays, I would be able to tell that in this particular case, there is not much going on in terms of chromatin accessibility or transcription factor binding as indicated by these two chip nexus datasets. Also, there is some degree of nucleosomal occupancy. So at this point, I could push the correlative analysis of these data sets more, but I wouldn't get much further than that. So what we do is single molecule footprinting, which works like this. It employs an exogenous methyltransferase enzyme to methylate cytosines that are accessible in the GPC context and leaves unmethylated cytosines that are protected by the occupancy of a nucleosome or also the binding of a transcription factor. So this is for the footprinting part. The single molecule part of the assay comes from the sequencing technology that we choose. In the case of my project, it was traditional Illumina bisulfite sequencing. And thanks to the chemical conversion of bisulfite, the methylation mark is converted into primary sequence, which means that each footprinted molecule, each sequenced molecule, then harbors the continuous accessibility information at the region in a binary form. It is by aggregating molecules by similarities of their footprinting status, of their accessibility status, that we get to be quantitative about this. So formally, single molecule footprinting quantifies chromatin accessibility at the single molecule level. And of this, I want to highlight two aspects. One is that because we do sequence molecules regardless of their accessibility status, we get to estimate the frequency of particular molecular chromatin states in a cell population, in a homogeneous cell population. So we do make our measurements in frequencies rather than enrichment. And we do so with a fairly high spatial resolution. We have on average, a measurement, a cytosine measured every 7 base pairs, which allows the pretty detailed dissection and profiling of trees in the mouse genome. So if I go back to this composite OCT4 and SOX2 binding site, this is what it looks like through the lens of single molecule footprinting. So I'm highlighting here a 500 base pair wide region surrounding this binding site. And the way that you can interpret the single molecule footprinting signal, which in this particular plot is shown at the average level, is by occupancy. So the higher the signal, the more occupied is each cytosine that we measure. And what I can conclude from here is that in 63% of the cell population, the binding site, which again is inactive and not bound by transcription factor, is accessible in 27% of the population, or inversely is occupied in 63%. So the most rich way, the most information-rich way that we can go about with single molecule footprinting is to do an analysis at the single molecule level. And for that, I've developed a novel computational method called footprint charter, which quantifies indeed the heterogeneity of chromatin states at the single molecule level. So formally, footprint charter performs unsupervised partitioning of molecules and detects in a data-driven way footprints for transcription factors and nucleosomes along molecules. So in practice, what this means is that it interprets the binary methylation footprinting signal into either of 3 biological states, either TF-bound, accessible, or nucleosome occupied. So to exemplify this, we can look at this cartoonish, very simplistic example of a tree that harbors a single TF-DNA binding event. And at this locus, I can imagine that we can find different molecular states, one of which could be found, for example, in the first 5 molecules, and harbors a footprint for the transcription factor, as well as to flanking nucleosomes in light blue. A second state could harbor, for example, the same nucleosomal footprints, but no TF footprint, hence a wider stretch of chromatin accessibility, and so forth. So for this particular talk, I will focus on 2 specific metrics that can be extrapolated from footprint charter. for each TF binding event. One is the frequency of chromatin accessibility across the cell population, and this will include the molecular states with and without TF footprints, as long as they don't include nucleosomal footprints, and the width of the accessible stretch that overlaps the binding sites. So pretty different in the nucleosome depleted region, the width of the nucleosome depleted region. So if I now go back to the same OCT4 and SOX2 binding site, what I can show you is that the raw single molecule footprinting signal looks like this, which can be read as follows. This heat map has one column per cytosine, which corresponds to a dot in the scatter, and one row per molecule, of which we have 674. Each cell in this heat map is colored by its occupancy status, so black for occupied and gray for accessible. And after the footprint charter state interpretation, this is what the same locus looks like. So again, one row per molecule. And the interesting conclusion that we can draw in this particular case is that this 27% of chromatin accessibility that we could already detect at the bulk level can be entirely explained by linker DNA. So basically, you can observe that in this 27% of the molecules in the cell population, accessibility is never wider than 92 base pairs, and it's flanked always by nucleosomes. So that's really pretty interesting to observe how an inactive locus can be accessible in 1/4 of the cell population purely because of the statistical positioning of linker DNA. Now, by contrast, I want to show you an example of an active element. This particular one is an active enhancer for which I can already detect an important level of chromatin accessibility and TF binding. And that through the lenses of single molecule footprinting and footprint charter looks accessible in 45% of the cell population with a width of accessibility which is compatible with regulatory activities. And the same goes for an active promoter, which is even more accessible and similarly wide open. So these kind of metrics are maintained across the genome. So promoters in mouse embryonic stem cells are very frequently accessible. You can see that the distribution is shifted quite up, and whereas enhancers show a much broader variation, indicating that for enhancers, TF nucleosome competition is quite a prominent is quite prominent at this crease. And the same can be observed by looking at distribution of widths at the same elements when promoters do display almost entirely accessibility widths compatible with regulatory activities, whereas enhancers also show shorter stretches of accessibility which are compatible with linker DNA, which is the same that we observe, which is basically only that we the only thing that we observe at inactive elements. Now I can zoom in a bit more into how individual transcription factors associate with different levels of chromatin accessibility. And transcription factors that are associated with that are frequently associated with promoters, such as NRF1 and FYA and BAMP, look very frequently accessible across the cell population, whereas transcription factors that are more associated with enhancers, such as OX4 and SOX2, show more intermediate levels, which, but also a wider spread, which indicates that there is some pretty important contextual determinants of the specific levels of TF nucleosome competition at this loci. As a matter of fact, if I unpack what happens around KLF4 binding sites, if I segregate crees that have an increasing number of KLF4 motifs, so 1, 2, 3, or more than 3 binding sites for KLF4, then these crees tend to be more accessible and with wider levels of chromatin accessibility. The same is true for an NRF1, albeit with a lower dynamic range due to the fact that these elements tend to be already quite accessible to start with. And of this, I would like to show you a couple of examples. So for example, we have a cree that harbors a single KLF4 binding event, which is accessible in 53% of the cell population, accompanied by a KLF4 cluster, which is accessible now in 68% of the cell population. Similarly, going from 1 to 2 NRF1 motifs also increases the fraction of the molecules in the cell population that show chromatin accessibility. Now, all of that I've been showing you has been derived by doing correlative analysis across the mouse genome. But to truly assign causality to the transcription factors, to specific transcription factor instances for the maintenance of chromatin accessibility, we've decided to perturb TF binding systematically across the mouse genome. And we've decided to do so by leveraging the natural genetic variation between different mice species. These species have been crossed to obtain F1 lines, where the idea is that by having both genomes in the same nuclear environment, we can observe that in some instances, this genetic variation will impose detectable changes in TF binding affinity, which occasionally translates to changes in the interactions between the transcription factors and their impaired ability to displace nucleosomes. So I want to show you one example of a pretty predictable transcription factor in the sense that in this particular transcription factor, CTCF does respond very predictably to perturbations in its binding affinity. In this particular case, we can observe that at the reference allele on the black 6 allele, there is a pretty clear footprint for over the CTCF binding site. However, the changing binding affinity imposed by the genetic variation at the binding site completely kills this footprint, which can already be observed at the bulk level. Even more clear is this at the single molecule level, where the TF footprint is completely abolished and the levels of chromatin accessibility, the frequency of chromatin accessibility is dramatically reduced. So, as I said, CTCF responds very predictably to changes in binding affinity, and this is true across the genome. So, the changes in chromatin accessibility frequencies are pretty unidirectional upon loss of function. However, this is really not true for the vast majority of the remaining transcription factors, where in most genomic instances, losing TF binding doesn't lead to a detectable reduction in chromatin accessibility. However, if I do focus on the instances where there is a statistically detectable loss in TF in chromatin accessibility, such as in this case, then this has a consequence. So in this particular case, I'm looking at a cree that harbors two KLF4 binding sites, one of which is perturbed by the natural genetic variation between these and my species, and this induces a reduction in 26% of chromatin accessibility across the population. And interestingly, this is accompanied by a reduction of transcriptional activity associated with this cree. And this same fact is maintained across the genome, where regulatory elements which are associated with an increasing reduction on chromatin accessibility are also associated with an increasing reduction in transcriptional output. And this is really interesting because we are really talking about reductions in chromatin accessibility which are really not complete. So in this particular case, we are really dealing with 26% of reduction in chromatin accessibility, which leads to, which has a transcriptional consequence, which means that cells really manage to, really can use, really can regulate TF nucleosome competition in order to fine-tune transcription overall. And with this, I would like to summarize and conclude. So I've shown you how through single molecule footprinting and footprint charter, we can profile the heterogeneity of chromatin states across a homogeneous cell population, and that KLF4 and NRF1 are examples of transcription factors which do contribute incrementally to chromatin accessibility. At the same time, transcription factor redundancy is a pervasive feature at CRIS, indicating that indeed CRIS are quite robust in their maintenance of chromatin accessibility. However, when there is an influence, then this tends to propagate downstream in the regulatory network. And with this, I would like to thank my supervisors, Arno Krebs and Judith Zaug, and all the members of both labs, which have contributed in very many ways to this project, as well as our external collaborators. And yeah, with this, I want to thank you all for the attention and eager to answer questions.
If you have any questions, please feel free to type them in the Q&A box or raise your hand so we can unmute you. So you got one question in Q&A box already. So great talk. I was wondering how footprint chatter causes TF binding. Is it just width of the inaccessible footprint, or does it also require no motif presence?
That's actually a very great question, and I always love talking about the technical details of my tools. So it's the detection of a TF footprint can happen without necessarily annotating the footprint. As a matter of fact, it is a matter of width. However, I do need a TF annotation to carry footprints in downstream analysis. So in practice, I do want to annotate this TF footprint with motifs, and I do want to validate them with also in vivo evidence of binding. So I always use orthogonal methods such as chip sequencing and chip nexus to be confident that the footprints that I'm looking at are indeed for the transcription factors that I'm annotating.
Well, thank you. You have a lot of questions. And so another question is, this was for transcription factors that are operating as activators. What about the repressors? In particular, have you looked at the transposon elements repressed via heterochromatin, HP1 or H3K9 trimethylation? Can you see transcription factor, not transcription factor binding? Yeah.
Oh, that's actually a pretty cool question. So we haven't explicitly looked at repressors. I have seen some anecdotic transcriptional repressors associated whose losses associated with a slight reduction in a slight increase then in chromatin accessibility. But we should totally bring this down to the transcriptional level. Yeah, that's such an obvious thing to look at. Yeah, thanks for the suggestion.
All right, thank you for answering that question. And then your next question is, first of all, very beautiful data. And then does the fraction of accessibility predict transcription factor occupancy or vice versa? Can your data answer this question?
So yeah, another great question. So in terms of predictability, I say it's a bit of a difficult question to I mean, it's a bit of a difficult thing to assess because there is also a few technical things in between. But overall, I can say that there is heterogeneity. So and I think it's also a bit of a way, the way that our understanding is limited of these biological entities, right? Because we are really educated to think about it in static terms, right? So a transcription factor binds and it creates accessibility, and that should be it, right? But in truth, the dynamics of transcription factor occupancy at crease really vary from TF to TF. So that's also a bit reflected in our data. So we do detect best and the transcription factors with longer residence time. So you've seen CTCF and NRF1, for example. REST also looks very importantly bound at regions. And these then tend to trace more with the then fractions of chromatin accessibility. Then there are transcription factors which we have a bit of a harder time detecting as bound. And this is partially due, in our opinion, to the residence time of transcription factors. Also, the fact that crease very often harbor very low affinity motifs, which are, for all practical purposes, really difficult to detect and to work with. And in principle, this should also leave footprints if they're functional. So yeah, I would say that it's a very intricate question, but I would be very happy to discuss for a couple of hours about this.
Thank you for answering all the questions. And again, if you have questions, feel free to type in Q&A box, and I'm sure Guido will be happy to type the answers in the second session. of the seminar. Thank you, Guido, for the wonderful talk.